more important and more practical, more real-life application here. Okay, so still animal cell, but now it's about how to really make the transgenic animal cell and then what to do with it. So first, you again need to know how to transform the DNA, how to transform the animal cell, how to put the foreign DNA into the animal cell. But again, go through the step again. For making transgenic organism, or for doing doing the recombinant stuff on the organism, you have to identify the gene you want to put into another organism because they give you some features that you want, like producing a new protein, making them resistant to everything. So identify the gene you want, that give you the feature, and then isolate the gene you want, and then find a way to put it into another organism. Here we call it transformation. So make sure you know the transformation technique we have. And then you have to check, after transformation, check whether the DNA are really inside. So you have to be able to do selection, screening, detection, something like that. And then you can make use of this transgenic organism. Okay, so when we talk about doing this in animal, again, transformation, we can do some physical way, electroporation. We can do again some heat softening, chemical thing. We can also have something more animal cell specific, like we can make use of the feature of the cell membrane. So we can do something called liposuction. We just bring something close to the animal cell and they will accept it because they have the similar structure of cell membrane. So they will fuse together, releasing the content into the animal cell. Okay, so now we really want to produce a transgenic animal, which is called transgenesis. And if we use a transgenic mice, as an example, here are the steps, important step of making a transgenic mice. Okay, so we can actually use other example. We can make transgenic sheep, we can make transgenic monkey, but mice is like the most used one. So make sure you know all the steps, all the key steps on how to do so. So at least you have to be able to tell all of this. Better if you can describe the detail of each of these steps. So we start with, let me see, where are we? Okay, so we start with building the transgene. So transgene is the gene you want to put into another organism. So it can be some natural gene that you just cut it out from another cell and put it into this organism, or it can be artificially constructed. Okay, we can make gene already. We can put up the nucleotide in the lab by ourselves, fully design a gene that is made in the lab. No matter which type, the transgene is something you want to put into the organism. So you have to make it out first. You have to consider that you have to add some promoter, because with the promoter, then the genes can be turn on and turn off the expression under control and turn on stronger when needed. So we have to consider putting a promoter. You have to consider putting a reporter gene. So later you can do screening. So when you make the transgene, you have to consider all this. And then you have to collect the fertilized oocytes because they are animal. You cannot just do the plant thing. You cannot just take a cell, put the DNA inside, and then go it into a new plant. You have to start with something like sexual fertilization. So you collect the fertilized oocytes from a mother mouse. Okay, so make sure how to do so. You get it, you make them, you make it, you stimulate it to produce egg fertilizer, but then you have to remove the original DNA materials because you want to put in yours. So you have to, at some point, you have to inject the DNA into the polynuclear, and then you culture it, so some step like this. Okay, so you collect fertilize the oocyte, and then you have polynuclear injection of your transgene, and then you transplant the embryo. Now you have a embryo, the fertilized egg developed into an embryo with your transgene inside. You put it into foster mothers, okay? Because you cannot just go into a baby mouse in your lab. So you put it back into a living mouse, which is a foster mothers. And these surrogate mothers will give birth of your baby mouse, and you have to again check which baby mouse have the transgene. Okay, may not all of them, some of them. So you have to have some way to check. We now we mainly look at their fur color, whether they have some pattern. Okay, because when we put the gene will give them some color on their fur, and they will be different from non-transformed baby mouse. So make sure you can tell the identification. And then, of course, you can always further confirm by doing, again, the PCR, checking the genome of the baby mouse, the whole genome, to find whether the new gene are there, blah, blah, blah. And so if you get the transgenic mouse, you have to further establish the transgenic line, okay? You crossbreed them, you breed them with some other mouse so that the new genes are stably established in the generation. Okay, so make sure you know all the key steps here about making a transgenic mouse as an example of making transgenic animal. And we also have something called the lockout mouse that we kick out some of the gene, and then we can check whether we can check like the gene function, we can turn it off, okay? We can also add some other genes, so have some idea on what is so-called lockout mouse, how to do so, and what is the application of this. And we also have gene targeting. Okay, so gene targeting is also another way to produce a transgenic mouse. So apart from doing the whole step, using the baby mouse, the foster mother, blah, 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 we can also do gene targeting to change the gene in an adult mouse. So you don't have to raise it, or you can still do it in an embryo and raise it. Okay, so in gene targeting, there is a concept quite important, which is related to this homologous recombination. Okay, so you have to know about what is so-called a homologous recombination, and I think this diagram can help you understand it. Okay, so basically we are trying to modify the gene in an animal. So how to target the gene to be modified and how to really modify it is by doing this homologous recombination. Okay, so we bring the new gene very close to the original DNA of the organism, and then we design the gene. Okay, this is designed by us, so we have to design some portion of it to be complementary with the original genome DNA, and this is so-called the homology arms. Okay, because they're complementary, then they will align together, and then the middle part is the gene we want to change. We want to swap it, so they will do this step is called the homologous recombination. So they will swap the gene with each other, and then the target gene here, because they are swapped, so they are swapped out, and the target gene will be removed. Okay, at the same time, so this is the resultant genome. You have to be able to screen whether they are really changed like that. So you add some marker. For example, you can have a positive selectable marker, and this one is something that will light up, that will give you some light. And you also have something called a negative selectable marker, because apart from homologous recombination, it can be also directly inserted into the genome. Okay, so then that is not what we want, because it is just inserted in the middle, but the target gene is still there, somewhere out there. And so you can, by having the negative selectable marker, if it is really done by homologous recombination, the negative marker won't go into the genome. But if it is by random integration, they just insert it into some point, the negative marker will also get in, so they can tell you if it is present in the genome. You can tell that actually you failed because you are not doing homologous recombination. Okay, so this is the key idea on how to do gene targeting, how to use gene targeting to make transgenic animals. So make sure you know about the concept here. Homologous recombination, what is it? What is leader? We need positive marker, negative marker, how does it work? So you can try to draw it, and then you can understand it more easily. And also watch the video. Okay, so some more about the gene targeting. Yeah, still about screening, checking for the transgenic animal whether successful or not. But still they have some limitation, and that's why here we come the most powerful tool, the CRISPR-Cas9 system. Okay, so the CRISPR-Cas9 system is like the most powerful tools to modify or editing gene nowadays. So it is like the number one thing we have today. So make sure you know what is a CRISPR-Cas system. How does it work? The components involved. Of course, we have the CRISPR-Cas9, the CRISPR-associated protein 9, the Cas9 protein. We have the CRISPR RNAs. We have the tracer RNA. Okay, so we have some detail here about the process, about the components, about how they identify a certain gene, certain parts of the genome very specifically, and then they can lock themselves there, and then they can cut something out, make a cut there very specifically. So make sure you know about how all this work, the components involved, and then after making a cut, you are modifying the gene. Then of course you have to be able to put something new back there. You can cut something out totally, or you can make a cut and then put something new in. So make sure you know how all this work. There is also something similar to the homologous recombination involved here. Okay, so if it is successful, we can do it by the so-called in the mutation. We can add something, we can delete something, we can also manipulate something, depends on how it works in your system. Okay, so at least you should be able to use this diagram, because it is like the very simplified version of the whole thing on CRISPR-Cas9. How it works: we have a protein, we have a guide RNA to identify the target region, and then they will attach to the target region of the DNA through the guide RNA, and the CRISPR-9 RNA complex make the cut. This is a double strand cut, and then so it break up the DNA, give you the gap that you can put some new DNA in. Okay, so this is like the most simple explanation here, but you better be more familiar with the whole thing. Okay, so you can tell the whole story. Okay, look at some video, look at the passage here. Okay, so we now have we talk about how to produce transgenic mice. We talk about gene targeting. We talk about CRISPR-Cas9 system. So these are the some most important tools and techniques in doing transgenic animal technologies. And then we also have animal cloning. We can clone some shape. Okay, still under development, but we are having improvement now, and some practical use. Okay, so again, no matter we talk about bacteria, plant, or animal, always be able to tell some example of the practical use, how we are applying these tools and techniques in real-life application. So for transgenic animal, of course, they can help us to fight disease. Okay. So make sure you know some example how they can help us to fight the disease. We can modify some animal that are spreading the disease, spreading the pathogens, blah blah blah. Animal are also food to us, so it can also help us to raise our food production, to improve the hunger problem, even to remove some allergens in food, so people can have more option of their food choices, and some other use, blah blah blah, something like that. So make sure to be able to tell some example here. And